All right, grade 12s, I'm going to do a quick take-up of everything on page 49, 349 that I assigned you. A lot of these answers here are things that you could look up, and I'm just going to give you some guidelines. So a large portion of the human genome consists of non-coding introns. Hypothesize some reasons that may explain this. So you have to remember one thing about the textbook. It is limited in its information and it's dated at this point. So I just took some time and looked up introns to see if there's extra information on them, and there absolutely is. So what they're saying now is that non-coding introns are, there is coding in introns, okay? So there's a lot of information in the intron itself that actually controls how the RNA is made, okay? So keep that in mind. But to answer the question, a large portion of the human genome consists of these introns. Hypothesize some reasons that might explain this. So a couple reasons that I just found are they suspect that maybe we went through bottlenecks and the introns that were represented, or any organism that has it, could have gone through a bottleneck, and the introns that were represented are the ones that we have now, okay? So that's one reason that we could have hung on to these introns, okay? Not a great reason, but it's a suggestion out there. Question 3 says, what is a pseudogene and how might a pseudogene arise within a genome? So a pseudogene is a gene that is in the DNA that no longer serves a purpose. So it's like a vestigial gene, right? And so it doesn't have a function. So I just wrote down here in my little words here, it's non-functional. And one reason that they think that they could still be around is because of mutations. All right, so that's one example, just one example. They're also left over from remnants from evolution, so that's a better example, okay? And I just thought I'd bring mutations to light because I was looking up pseudogenes, and they did mention, different research facilities mention that when people come in with gastric cancer, so cancer of the stomach, they have a lot of mutations in their pseudogenes, all right? So that's one example there for you, that's real and current. Epigenetics, had you guys been in class, I would have had you do seminar presentations. So some of you are going, oh, thank God we're not there. But these guys, this is one of the topics I have students do. So I just wanted to at least introduce you to epigenetics. Epigenetics is this. Different genes are turned on in different situations. So I hope that you did a little bit of research here because this is really interesting stuff. But the example I'm just going to share with you is stem cells. So give stem cells different environments and they will turn on in different environments and they'll turn their genes on, okay? So that's an example that everybody has gone through with their stem cells, but there are so many different examples of epigenetics, okay? And I hope you came across some really interesting ones. And again, epigenetics just means genes are going to be turned on in different environments, okay? So not every environment is going to have those genes turned on. For example, the agouti mice and the agouti gene, okay, that we looked at a week or so ago. 7 says, describe the ramifications if a mutation were found in each of the following. So to the best of my knowledge, again, your textbook is limited in the amount of information it gave you, so you'd have to do a little bit of research, possibly. If a mutation was found in an intron, introns control RNA, so it could be detrimental to the cell, okay, and detrimental to the DNA. So that's what I would say. It would harm protein synthesis. The exon is coding DNA, so it would definitely be harmful. All right. Telomeres would influence when a cell goes into cell senescence. Okay. So they're just looking for some kind of a hypothesis here. So if you're looking at the telomere region and we're talking about a mutation in the repeating sections there of DNA, then we would probably say that it's going to influence aging and cell senescence. Sign, unknown up to this point. I did not research that. So if you want to research that one, if you're interested in this stuff, because I exposed you to this because I figured some of you guys are going to find this cool. Okay, and it is something I would normally give people to do anyway. But if you want to look that up, you would probably find some very interesting research up to date. A pseudogene, I'm going to say that there's probably no ramifications to a mutation in a pseudogene because we're not using the pseudogene anymore. And variable number tandem repeats, I would have to say that there's not really any ramification, but that's just knowledge up to like 2019. So who knows what's been found in the last 6 months. Okay, so I haven't done any research on these guys this morning. So I would suggest you look that up if you're interested in it. I will not be testing you on this. But again, if your interest is there, there's so much information on this and it's so interesting. And then lastly, I had you do some research on viral DNA. So some of our human DNA consists of viral DNA that entered genomes hundreds of millions of years ago. So when we got an infection, you know this now, and this is why we're shutting everything down because of a virus. What they do is they insert their DNA into our DNA in some cases. And so we have in our DNA so many copies of viruses that we've encountered over all of the evolution of humanity. And so that's what I wanted you to do there. Just a little look up on the internet, other sources, research the viral DNA in the human genome, where they think it came from. So that's a personal thing to do for you. If you're interested, again, and if you've got time and you're bored, I would suggest doing that if you think that you're going into genetics, because there's so much interesting information on that. Okay, so I just left you with that. In addition to that, it was either before, or here it is. If this is really your thing, okay, I didn't assign it, I thought about putting it up there. I'm just going to recommend you read page 350 to 352. It's so important as to what's going on right now. But what I wanted you to just hear the term, I wanted you to hear reverse transcriptase. So when you think reverse, reverse means backward. Transcriptase, think transcription. So when we go through transcription, we take a DNA copy and we make it into an RNA copy, right? We're talking protein synthesis here. What viruses do is they take their RNA and they make it into a DNA copy. So they're reverse transcriptioning, all right? Reverse transcriptase is the enzyme that does that for them. And so once they do that, they can enter our genetic code, and that's how they can insert their DNA into us, okay? Because we don't recognize it because it's now in a DNA format. So they take RNA and make it into DNA. So in light of COVID-19 and everything that's going on, I think you should read 350 to 352 if you're interested in furthering your education, okay? It's a great thing to read. Take care.